One of the biggest concerns with gene editing tools like CRISPR is the same as one of the biggest reasons why drug candidates fail, which is the same as one of the reasons why your PCR reaction might fail in the laboratory. I'm talking about off-target activity. So we talk about a target being the thing that a molecule is designed to bind and have some effect on. Or maybe it's not designed to do that, but that's what scientists think that it does. They predict it does, they find it through some screen or something, then they think that it affects this molecule, and they have some great hypothesis about what it's going to do when it binds to that molecule. And maybe it does that thing, but then it also does some other stuff. And so an example of this is going to be antibiotics that target bacterial ribosomes. They can actually also have off-target activity on our mitochondrial ribosomes. So inside those powerhouses of our cells, there are mitochondrial ribosomes that are similar looking to the bacterial ribosomes. And so the antibiotics that bind to and inhibit bacterial ribosomes can also bind to mitochondrial ribosomes, and that can cause side effects. And that's just one example of off-target effects. Off-target effects are often going to be reasons why you get side effects from various treatments and adverse events, various things that happen with drugs that are on the market, as well as keeping drugs from reaching the market. But at least with those drugs and stuff, you stop taking the drug and normally all's okay. The thing with gene editing is that these edits are going to be, therefore, there's a lot of concern about kind of having unwanted edits to the genome. Before I get too into kind of some of the strategies that scientists use in order to avoid off-target effects, in order to detect off-target effects, and to work around off-target effects, let's first talk about go back to the difference between off-target and on-target. So both of these can cause kind of bad effects, and both of them can cause kind of good effects. In the case of the on-target, so on-target means that the molecule is doing what you think it should do. So it's binding to the molecule you think it should. So maybe you're trying to inhibit that bacterial ribosome, and it's binding to the bacterial ribosome and inhibiting translation. That would be on-target activity. Maybe your CRISPR guide, so that RNA that directs the CRISPR-Cas machinery to a specific site in order to edit it, maybe that guide is actually binding to that sequence that you want it to edit. All of these would be on-target. Now, sometimes the on-target activity is going to kind of be overkill, and so say you are trying to inhibit an overactive kinase in a tumor cell, but maybe it also inhibits the activity in the normal cells around it, and that activity might be really important for the cellular function. And so that activity would be on target, but it would be having these kind of side effects potentially for the patient. In the case of on-target activity as well, you could have consequences that are beyond what you think the consequences will be. Often we talk about pleiotropic effects, where basically pleiotropy means that something's having multiple effects. And so the term comes from genetics and things, when a mutation in one gene can cause a variety of different effects because a single protein, say, is associated with a lot of different pathways, or it's doing a lot of different things. And so if you mess with that one thing, you create these kind of ripple effects throughout. Just like a mutation in a gene can do that, so can inhibiting the protein that the gene would make. And so you can get these kind of pleiotropic effects from an on-target activity, but you can also get them from off-target activity. And so off-target activity, remember, this comes from the molecule binding to and acting on something different than the kind of target molecule, the thing that you think it's going to bind to, you want it to bind to, you intend for it. Now, this is not always bad. So sometimes this is bad, and this causes various problems in side effects, adverse events, and we'll go into some of the ways that scientists try to kind of avoid these and work around these. But they can also sometimes be beneficial. And so sometimes these drugs are actually acting on multiple things, and you don't realize they're acting on multiple things, but by acting on multiple things, they're actually having a beneficial effect. And sometimes things are working on multiple things and you don't actually know the target. So this gets down to kind of phenotypic-based screening versus target-based screening or mechanism-based screening. And so basically what happens was that in the olden days, before people had all this technology to be able to work with individual molecules and figure out the molecular causes of things, they would do phenotypic screen. So phenotype, if you think like genotype versus phenotype, phenotype is what you see, genotype is the genetic root of what you see. So your genotype might dictate that you have brown eyes, and then your phenotype would be the brown eyes that you see. So scientists could basically add drugs. Maybe they have some sort of big library where they have a bunch of different drugs that they can screen against. And they have a cell-based model of some disease, and then they add this drug to all these different wells that are mimicking this disease condition. And then they look for a phenotypic effect. So maybe they look for the cells becoming normal looking or something, or they stop growing if it's a tumor cell model or something. And then they're like, okay, well, this is going to have this effect. We don't know the target. It might be targeting multiple things, but all's okay. And then sometimes maybe scientists go and they try to figure out the target, and maybe they do figure out the target, maybe they figure out there's multiple targets, maybe they think they figure out the target, but it's actually something else. Yes, that happens. And then maybe they are thinking it's this one thing, and then they follow up and they do all these things and they try to develop this drug program around this molecule they think is the target, and it's not actually the target. Again, we'll get into more of this in a minute. But bottom line is that off-target effects might not be bad, they might be beneficial, but often they're going to be bad. And so often they want to be avoided. And we've been talking about these small molecules, but off-target effects with gene editing are pretty much always bad. You do not want to be editing the wrong gene. And sometimes you don't even know if you are editing the wrong gene if you're not careful. Which is why there's so much concern about kind of off-target activity when it comes to gene editing, and why there have been so many developments to try to improve the specificity of the gene targeting, as well as efforts to detect these potential problems, detect these misedits before these are actually used. And so let's go back and talk more about kind of how scientists can do this. We'll start with the CRISPR because that's the context that you hear about off-target activity most these days. And so it's most significant because it's going to potentially have these permanent effects. And if you're doing these misedits, that can actually cause things like cancer potentially if scientists are not careful. And that was actually one of the reasons why early gene therapy treatments were kind of like stopped, was that they were using older techniques that were actually causing cancer because they were using these delivery techniques that were integrating into regions of the genome that were causing these problems. And so the technology has advanced a lot since then. It is a lot more specific, but there still is concern about off-target effects. To understand how scientists can predict and avoid these off-target effects, we need to go back and think about how CRISPR works. And so much more on this elsewhere, but CRISPR basically works by using a guide RNA that you design to target a specific site on DNA, say that has a faulty gene. And then you have a Cas protein, and this Cas protein binds to that guide RNA, and then that binds to the site and it cuts it, and then the editing is made. If you include like additional information, then it can incorporate, it can kind of like swap out regions of DNA through homologous recombination. It can just make a cut and then rely on the cell to fix it, which typically leads to a knockout. So basically you just inactivate the gene. There are newer techniques like base editing where you just make single nucleotide changes, and prime editing where you kind of put in the new sequence without having to rely on homologous recombination. And so there are various methods, and some of these don't even involve cutting the DNA, but the traditional CRISPR-Cas involves actually cutting the DNA. And cutting that DNA is then going to leave a mark, and that mark is actually something that scientists can detect. And so that is one of the ways that scientists detect off-target activity when they're trying to design these guides to use. And so when we talk about using these, a lot of the attention is often on using them in the clinic, but a lot of the actual use of these is in the lab. So scientists use these CRISPR-Cas systems in order to study disease mechanisms, in order to study potential treatments, in order to study all of these sorts of things. And off-target activity, although it might not be harmful to a person in that case, it could be harmful to society if you are wasting all this money and stuff trying to track down this various target, and you think you have this activity, but you're actually targeting the wrong gene. And it could potentially be harmful to a patient then in the future because you are going after this like false hit when it's really something else that's going on. And so it's really important that scientists validate their models and validate things in multiple different techniques and kind of like orthogonal techniques. And so you're trying to kind of show things with different experimental techniques. But it's really a problem if it was then to go into a patient when you have these sorts of problems. So the key thing is to avoid these problems in the first place. And then if they detect them early on before you actually use them, or you use these cells, maybe these CRISPR knockout cells that are actually knocking out the wrong gene, all these various things, early detection is key. And so those double-stranded breaks serve ways that scientists can then use experimental techniques that are going to sequence the region where those breaks occurred, and that is then going to show them where there are potential off targets that they need to keep an eye out for, and that potentially they can design around. So at the design stage, before scientists even go and use those sequencing-based techniques in order to find the potential cut sites, what they often do is a computational screen. And so they take the sequence of the region that they want to edit, and then they work computationally to try to design a guide RNA that's only going to bind there, and it has low off-target predicted activity. And so if they see that it's similar to a sequence over here, well, then maybe they'll try to alter kind of like the site where the guide binds and this sort of thing in order to make it so that it's not going to have that off-target activity. And if they recognize those off-targets through some sequencing method where before they actually do the real edits, they just go and see where that Cas protein is binding and where it's going to be having effects, then they can modify their guide so that it potentially won't target those. And if they can't modify their guide, they can at least screen for those potential off-target edits when they do those kind of like real edits. Also, if you have this non-specific activity, then it's going to be reducing the efficiency of the on-target activity. And so the computational screening will help for that, and then also trying to increase the specificity of the guide through chemical modifications. So maybe they kind of add these chemical modifications to the guide that makes it so it's pickier and will only bind to that really specific site. You can also improve the specificity of kind of the Cas enzyme. So we're talking about the guide, but the enzyme is also really important. So you can improve the specificity of that. You can use kind of nicases where you have two Cas enzymes that use two guides, so you get kind of dual level of specificity. You can also use these enzymes that don't actually cut so that they'll potentially do less damage if you do have off-target activity. And when scientists think that they actually have a CRISPR knockout line, then they should do things like actually testing and making sure it's not making that protein, sequencing it, all these various things. And then again, if you want to go and then apply these, say you want to do this ex vivo, so you take, say, the stem cells out of a patient and edit those, well, then you're going to want to sequence those and things, at least check those sites that you predicted would be those potential editing spots. If we're trying to introduce CRISPR in vivo, so actually in a person, you don't want it making edits that you don't want it to make. So scientists want to predict what off-target edits it might make, then redesign their system so that it doesn't make those before it actually gets widely used. And so again, there's a lot of development going into these, and so this is not to scare anybody off from gene editing, but it is important that you keep this in mind. This is one of the reasons why scientists aren't just going around willy-nilly and editing the genes of people all the time. In terms of pharmaceutical drugs and things, so if we're thinking about these target-based screens, and you often would have like a protein, or you have some sort of assay, and then you have these compound libraries that maybe you're screening against, and then maybe you find a hit. And so you're like, okay, so I know that this molecule is going to target this molecule. That would be on-target activity. But is it also going to bind to other things? And the other things it's most likely to bind are things that look like that molecule that is your target. And so often what you could do is you could have screens, then further screens, where you have a kind of a panel of similar looking proteins that you can then test for activity against those proteins. And so a common place this comes up is like kinases. And so kinases are proteins that phosphorylate other molecules, and these are really common in signaling pathways, which are really commonly mutated in cancers. And so kinases are a key target for a lot of these like anti-cancer compounds. But kinases are very similar looking, and so a single molecule that you think is going to be specific for this kinase might actually inhibit other kinases. And because scientists know that, then they can have this kind of like panel of these other kinases that are similar looking in order to be able to predict or detect if they have activity on those kinases as well. You could also have off-target activity though on things that you do not expect. And so that's why then there are kind of like phenotypic screens and various things like this to examine what off-target effects a molecule might have before it's taken further into clinical studies. So this would be kind of like more unbiased ways to kind of like see toxicological effects potentially, and then the scientists could follow up on those. There are also panels of proteins that are known to have really bad side effects if they are accidentally targeted, all these various things. And so scientists can kind of predict ahead of time what it might be binding, and then they can also go and look for what it actually is binding. If they find what it is binding, then what they can do is they can actually kind of try to design their molecule to be more specific for the target that they want. And so this might involve solving the structure with like X-ray crystallography, or maybe just using a predicted structure, doing molecular docking, trying to see how this molecule is binding to a specific location on that protein, and then trying to compare the structure of that to the structure of these other off-target proteins in order to kind of rationally design, using that structure, the molecule. And so maybe they'll make changes to parts of the molecule in order to try to improve the specificity. And then they need to make sure that those don't also kind of take away the activity. Also, remember that on-target activity isn't always great either, especially if it's too much or if it's in the wrong kind of tissue type. And so improved delivery of molecules, whether it be the guide RNA for CRISPR, whether it be these small molecules, you want to make sure that they're actually getting to where you want them and only where you want them. And so improving the delivery mechanisms for these is also a way that you can at least limit any off-target effects, as well as any on-target effects that cause too much activity or too much targeting, that sort of thing. So on-target, off-target, these can both cause bad things and they can both cause good things. The key thing is that it's important to know, and sometimes you don't know, which is bad. And especially when it comes to gene editing, it's really, really important that these guides are designed in a way that is going to be safe, it's going to be effective, it's going to be efficient, that the CRISPR-Cas machinery is going to be able to carry out these edits in a efficient way that is high fidelity, so it's not making errors you don't want. And this is important in the lab so that we don't waste money and effort and kind of have wrong hypotheses because we're thinking something does one thing and it does another, and it's bad in the clinic. I've been talking about the CRISPR, but you might come across something similar with your PCR reactions in the lab, so just a quick note about that. If you have off-target binding of your primers, so the little pieces of DNA that you use to specify a region of interest to be copied, what happens is that it can bind non-specifically to sequences that are similar, and then you kind of get multiple products. If you're doing something like qPCR, where you're not actually looking at the products, you're just kind of, oh, I see light that tells me double-stranded DNA is being made, then you could kind of like get a false positive if your primer is binding non-specifically somewhere. So similarly to how you could use computational tools for the guide, you can also use computational tools for primer making. And so like Primer Blast from NCBI, you can use those tools to try to predict offsites and better design your primers, lengthen your primers so you can increase the annealing temperature. All these various things are going to help you have better specificity. And then if you're doing something like qPCR, you're going to want to validate your results. Maybe use something like TACMan, which has a probe that's actually specific for the amplicon, so the thing being